So I did a short one-page cheat sheet for all of you as well. So I'm planning to do the same kind of summary after each of our main or bigger topics and subjects. And this is all about the prokaryotic de novo genome assembly. I hope you can see it now. It will be, of course, uploaded in the Google Drive, so you will find this document there, but it is going through all the steps. It is going through, it is also having all the command line commands with, I think, easy to follow format. Also, there are some tips for further things in brackets after the command line command. For example, if you have more CPUs or more memory than you can use, or if there is any option which should be used, is there as well. So I think this is quite useful and a good first step if you want to do your own genome assembly. As we discussed before, maybe you don't have to do the genome assembly just because it is already done. But as soon as you find something strange in your data, and there will always be something strange, there will always be something which seems like an incomplete genome or a contaminated genome or something like that, and then you can go back to your raw data. I don't know if you send your sequencing to the Sanger, if you have chance to get the FASTQ files. I think so. I only sent sequencings to a place where they only do the sequencing and sending the FASTQ file, but they don't do the assembly. But I think you always have the right, or you always have the chance to ask for the FASTQ files as well. And especially if you have a problem, then you can go through this whole pipeline step by step and check where is a potential problem in the data analysis. Okay, so what we will do today is just the last part of this, and then I will speak about a little bit more about the a kind of like a decision tree. If you have to do some kind of whole genome analysis, then what are the main points where you have to decide if you have to do A or B or C approach. And it's basically the main choices between de novo and realignment. And then on Thursday, we will go through a complete realignment pipeline. I think we only have chance to go through a prokaryotic realignment pipeline. Maybe I will create something like this for eukaryotic as well. The problem is that going through the eukaryotic one is just too much time. It's very, very long. Okay, so we did the mesh last time. So the mesh was the program that was our last successful program we were able to run last Thursday. And we weren't able to do the CheckM, but we will do. Are you ready, Joe, to finish what your task was last week?
I mean, do you really want me?
Yes, this was your topic, so please do it, and then I promise I won't disturb you anymore in the next 3 weeks or so, because I want everyone to contribute. But this was actually yours. Just it was my fault that you weren't able to complete. So the CheckM is installed. I sent you the how can you install in your own server if you want. It's not as easy, not as simple as it is there on the website. But now, for me and also for all of you, you have a separate environment for the CheckM. And as soon as you want to use the CheckM, before you want to use the CheckM, because of course, if you log in and try to run CheckM, it's not running because there is no CheckM found. So the first thing we have to do is to say conda activate py27. Of course, you can name this environment a bit more with a bit more obvious name. It's just named because it was the default, or it was the suggested name for the CheckM environment. If you say this, then you will see that the base prefix before your prompt will change to py27. And from here, there is a CheckM command. If you start, you can see that there is a lot of different things you can do with CheckM. So we will do one CheckM run, but I will only ask Joe to do the run, because even if one people is running and using all the CPUs, it is about 5-10 minutes. So I think it's the best to start running, and while it is running, I will tell you what it is doing, and then you can understand how this whole program is working. So now I only ask Joe to do the things. Go to your Scratch directory. Scratch. I will go into my own, and you should have your context-FASTA file in your folder. I don't think you have the directory bins. The reason why we should make a directory for CheckM is because it's usually we don't run CheckM on one assembly. You either run it on a metagenomics assembly where you have a lot of different files, or you are just getting back your multiple sequencing files, multiple isolate sequencing. It's very rare that you are sequencing just one colony at a time. You're usually doing more than one. And if you have all those config-FASTA files out of the de novo assembly, you can just put them into one directory and do the whole CheckM process on all of them with one command. So we, instead of doing it for multiple files, we will just do it for one file, but even that one file has to be in a folder. So if you don't have this bins folder, what I have, just please do it. So make mk dir bins. Of course, I don't do it because I have it. And just simply, if you have the bins directory, just copy your context-FASTA to your bins directory like this. Of course, anyone can do this now, and I'm just feel free to do it, but don't run the CheckM. We will. I don't know if we should follow the run. Let me think. Okay, we don't really have to see the output. Okay, I actually can show the output from my run, I think. Yes. Okay, so if this is done, then the command you have to run is there will be a lot of different CheckM commands we will run later on, but this is the main command we will first run. So your first CheckM workflow should be always this lineage workflow, which means that CheckM will check a lot of things, but also try to identify the taxonomy of your config or your config-FASTAs. It depends on how many different files you have. You only have one file at the moment, so you have to give the command, CheckM command, and then you have to give the sub command, which will be a workflow, which means actually if you go up to the CheckM website, it is a multiple sub commands after each other. Then we have to give a -x FASTA because in default CheckM is looking for .mfa files, which is the extension for multi-FASTA. In our case, we have context .fosta. So that's why we have to tell CheckM that it has to look for the extension. That's the -x extension fosta, minus T4. This is the number of threads, number of CPUs, minus reduced 3. If you don't have enough memory in your machine, then you have to use this option. The enough, according to the CheckM website, is about 9 gigabytes of RAM. So if you have less than 9, then you have to use the reduced 3. We have 24 gigabytes in this computer, but I'm not completely sure that we can do it without the reduced 3 option. And then, and it's faster if we are using this. And then this will be your directory where your contigs are, where your assemblies are, sorry, where the assembly FOSTA files are, and then you have to give an output folder name. And that's all. Tell me, Joe, if you started. You have started it. I can actually.
Yeah, I'm running it.
Okay, so it's running. So it will give you some output during the run, which is also the same thing that you can also see in later on in your CheckM output folder. So if you're looking at the CheckM output folder, you have two main things we will work. You don't have it yet, but this will be there when the CheckM finishes running. There will be a few folders with a lot of different files inside, which you don't really have to look into. They are not really in a human readable format. But the main thing what you have to work with is this lineage.ms file. Again, don't look into it. Just use it later on for the other subcommands. And you can see what happened during the CheckM log. And the reason why I want to show you the CheckM log is because I want to show you how the whole CheckM works. Because again, if you know how CheckM works, you know what are the pros and cons using CheckM. So, so the first thing what it does, and this is some probably where your run is at the moment, this first 2 minutes, is it has 43 single copy genes that it checks within your contigs FOSTA file. Of course, in this case, we only have one bin, one FOSTA file, but if you have more than one file in your bins directory, then it will do it for all of them. So what's the rationale behind only checking 43 genes? You know, in each of our prokaryotic genomes, we have at least 2,000 genes, or quite often we have at least 2,000 genes. The reason is that CheckM is only using 43 because these 43 genes are always single copy genes, or almost always single copy genes. And the other thing is they are also always there. So they are very basic genes which are there in all prokaryotic genomes. Of course, if you find 40 from 43, that means in one sense that you are missing 3, which is a bit less than 10%. So you can assume that your assembly is not complete. Let's say it's just 92 or 93% complete, because you found 92 or 93% of your target genes. And of course, if you have more genes to check, then you can predict the completeness of your assembly much better. But CheckM is only working with 43. I will tell you at the very end what is the alternative of CheckM in many ways nowadays. The reason why we are using CheckM is because even CheckM is requiring a lot of resources. The alternative we wouldn't be able to run on this server because it requires more than 100 gigabytes of RAM. So the first thing is that CheckM is not checking DNA sequence and looking for DNA sequence with BLAST. It is using HMM. Probably a few of you, or maybe all of you, already saw this thing, HMM. This is the abbreviation of hidden Markov model. And hidden Markov models are extremely useful if you want to find a pattern, especially if that pattern is not completely conserved. So let me give you an example. Let me show you something. So if you go up to the PFAM site, you don't have to do this. Let me just look at my screen, especially if it's working. It's not really working now. I think there is a European PFAM website as well. Okay. Let me just show you an image. I hope I will find, yes, an image here. So if you look at, for example, this image here, which is the PFAM or an HMM logo, it is called. So what you see here is that within this, probably it's a protein sequence, within the protein or within that domain from 64 to 81 position, these are the probabilities of different amino acids residues. And what you see is that, for example, in position 77, there are a few residues, a few letters here. They are very small, which means that this position is very not conserved. It's very unconserved. I don't know if there is any word like this, but you can have quite a lot of different residues in that position. But if you just go to the 76, as you can see here, there is a very high chance that you have a glycine here, or maybe a serine or an alanine or a threonine. So if you have this kind of profile for a gene, and now we are speaking about the protein sequence of the gene, then you can weigh the different positions by their conservation as well. So if you have a G here, that will be a very high score in finding a good hit. If you have a G here, that won't make any difference, probably in position 77 related to the final score. If you have a D in this position in 66, then it will be again, will score as a high score contribution to similarity. But also if you don't have some kind of residue, for example here, but you have that residue in your protein, that's very against the hit. So it's what I want to say is it's very different from for example, what you do with BLAST. In BLAST, especially if you do nucleotide BLAST, you are just looking for similar or not similar nucleotides. If you are doing protein BLAST, you are looking for similar amino acids or so-called, identical amino acids or similar-ish amino acids. If you have an leucine somewhere and you have an isoleucine in the same position in a different protein, they are very similar in characteristics to each other. But all of these things are governed by a substitution matrix, which gives you an idea that, okay, having an isoleucine instead of leucine is okay, but having an aspartic acid instead of an isoleucine is not okay. In this case, it doesn't depend on the amino acid itself. It is also true that in many positions, if you look at, for example, these 68, 69, 70 positions, the most probable amino acids are actually not very different from each other. They are all hydrophobic residues in this case, for example. And here again, you can see that D is here, which is an acidic, that's aspartic acid. there's an acidic residue, and also E is here, which is the glutamic acid. So in many cases, there are similarities in characteristics as well, but it's not just like that. The HMM logo and the HMM search is always taking the position-specific probabilities into account, the position-specific improbabilities as well. So if you don't have there, if you don't have it there anytime, then you shouldn't have it there in your target protein as well. Of course, building up these HMM logos or building up these HMM files, and we will speak about this later when we will discuss the similarity search. To build up this, you need a lot of known genes. So if you want to build up the DNA A bacterial gene, you need at least 100 or 200 or 1,000 genes and protein sequences from different organisms. And then you can extract all of these position-specific residue probability informations. And using that HMM logo or that HMM file, you can look for DNA A genes or translated DNA A protein sequences within your context. And that's what CheckM does. It's way much more sensitive, but also much more universal way of finding a gene than looking for nucleotide similarity. When you are looking for an antimicrobial resistance gene, the AMR genes are extremely conserved genes. Also in the nucleotide level, we find many cases when a gene that is 1,000 base pairs long, only having a few SNPs here and there. A few means that less than 5. Of course, if you have 955 nucleotides completely identical, it's very easy to find a gene with BLAST. But as soon as you have the same gene, but probably encoded by a very different DNA sequence, just because it was maybe evolved as a paralog, but encoding a very similar functional gene, then you have to go to the protein sequence and you have to go use the HMM search. So CheckM is looking for all these 43 genes and give you back a number or a few numbers about how many of these genes were found. How many of these genes were found in single copy? So the first thing, how many genes were found, will give you an idea on the completeness of your assembly. If something is missing, then you have to start thinking about maybe there was something wrong with your sequencing, there was something wrong with your DNA extraction, or there was just simply something wrong with your bioinformatics pipeline when you are at this point. So all of these things are very important. I think in most of the cases you have to do a DNA re-extraction and a resequencing if you don't have good enough assemblies. And I'm not pretty sure what is the good enough in this case, because I'm doing more metagenomics than single genome assembly. In metagenomics, I'm very happy if I have a 60-70% complete genome, because that's already a very good starting point. For example, finding out what is what's the genome is, so what's the bacteria is. But of course, it's not enough if you want to really characterize your genome. So if you sequence your colony because you want to characterize all the genes in it, you want to find metabolic pathways, and you want to find complete metabolic pathways within your genomes, then you have to aim for very complete, 99%, 98% complete genomes. The second thing is, how many of these 43 genes were found only once, or how many were found multiple times? How can it be that you have one colony, you are sequencing it, you are assembling it, and then a single copy, a known single copy gene is found in multiple copies? The reason most of the times is that your culturing wasn't pure enough. So if, for example, we had this case with my part2 student when we were analyzing Staphylococcus chromogenes genomes, which were cultured from milk, from cows with some kind of mastitis. It was probably a subclinical mastitis. So the milk was just simply put on a plate, and then colonies were picked up and sent to sequencing. And probably it wasn't, I mean, somehow the colony which was picked up was over or grown together with another colony. And we just simply sequenced more than one species. In a few cases, we sequenced two different genomes. I think in 1 or 2 cases, we actually sequenced 3 different genomes. So what you will see in this case is that, I have several duplicates. If you are sequencing multiple genomes very nicely, that means that probably all of these 43 genes will be there as duplicates. Okay, and then as soon as it finds the HMMs, it will extract the amino acid and the nucleotide sequence from these known genes, and using the nucleotide or the amino acid, I don't know exactly which one, I think much more the nucleotide sequence, to align those and compare those sequences to its database. So the CheckM has its own database. It is extremely outdated. It is from 2015, but that's the only available database for CheckM. I think you can expand CheckM database somehow, but at the moment, what you can download and what is downloaded for us is from 2015, and it will align. We create a multiple alignment for all these genes and compare them, and we'll calculate an average nucleotide identity or diversity or difference score. And that's where it will end. It will tell you which one is the closest gene set, what CheckM knows, and it will output that information for you, that I think this is this kind of genome. I think maybe it's finished now for you, Joe.
Yeah, it's done.
So you saw all these log files, and you also saw a very short table, a very simple summary table at the very end. It looks something like this. Let me just show you. I think it should look like something like this. So this is it?
Yep, it's the same.
Okay. So this is what you will get. So I reran the CheckM again, but instead of running CheckM with the lineage workflow, I only do the QA module, which is the quality assignment module for the CheckM. And I gave the CheckM output lineage MS, which was done during the lineage workflow, and also the output folder where all the other files are. And I don't have any idea what other files this used, but actually this is the same what you should have as the main output. And what you can see here is that there are some numbers here, and you can go and check the documentation, what are these, but I think the most important thing is here. So the completeness of this genome is extremely high, and there is some very few contamination. I think there is maybe a larger gene set which it also checks, and I think the number here at these 2, it means that there were 2 genes that were found in 2 copies, but I can imagine that this is just simply because it was a real gene duplication. So I think in most of the cases, if you want to extract some information from your assembly, you will use this QA, but not in this very simple form. So if you go up on the CheckM website, and this will be in the cheat sheet, so don't worry if you don't follow me at this moment, because this is all in this Word document. I hope. Yeah, so it's here. The URL is here for the CheckM Wikipedia. And if you go on the QA here, then what you can see is, it's not really here. It is this QA that you can ask many different types of statistics on your assembly. So if you say -0, -01 should be exactly the same what we saw now. So that's the default output. Yes, it is. But if you want to see a bit more, then you can go for the -02 to have some extended summary. And for example, in this one, because in this short summary, you don't really have anything about your assembly quality. You have information on your genome quality. You see that it is probably very complete, but you don't really see how fragmented it is. So how many contigs you have. So if you go on -02, then you will have way much more columns, and now it's, of course, it's a bit more messy here, but you can always extract these informations and just copy and paste into Excel or something like that, or just extract that column that you know is important for you. So for example, here you can see that there is a column for genome size, which is probably this one. We also have, of course, the same thing, that it's probably a Staphylococcus species. There are the number of contigs here. So as you can see, it's a bit fragmented. The N50 is here, which is a very important kind of statistics for saying how good your assembly is. So N50 is actually the length of that contig, of the shortest contig, which, if you add up all the long contigs in your assembly, this is the last one that has to be added to have half of your genome size. It's a bit strange, but it's a good statistical property for your assemblies. Of course, when we do metagenomics, for example, we are very far from this, very far from these numbers. If you have only one contig because you were able to assemble a whole genome, then of course this will be the length of that one contig. And also there are many other things here, like the GC content. So this will be the main thing what you really want to see. If you have some suspicion that you are sequencing more than one thing, for example, because your contamination level is high here, or your strain heterogeneity is high here, then you have a specific option for this output option. Yeah, here are the gene loss or duplication assembly errors. The -07, I think, is good. maybe the -06 as well. I won't go through all of these options. I just really hope that you will chance to, for example, have your own assembly. Just take one of your FASTQ files. If you don't have a FASTQ file, I can always give you one. I can also give you a FASTQ file which will produce a mixed assembly. So if you want to see how it looks like when you are sequencing something which is not pure, which is contaminated with something else, I'm also very happy to give you something like that. And I ask everyone to log in at least once and do this whole pipeline yourself and see what is happening in different points, and especially try all of these options. They are very fast. As soon as you run the CheckM, the main CheckM lineage workflow, all of these QA comments will work from the already generated files, and you just have to go through and see what you can see. So I think that would be the best for all of you. I think I will go to the next stage now, because I want to discuss at least 2 more programs, and then we can also have some kind of wrap up at the very end of the de novo assembly pipeline. So the next thing, which you probably want to do because of multiple purposes, is to annotate the genes. So I again ask only Joe to do this with me. So let's go back to our home directory and install PROKKA. So let's first Google PROKKA. We are looking for this GitHub repository, the rapid prokaryotic genome ... Before we go into the details and the installation, I don't know if I told you last time, but this guy is our God. According to Ibrahim, he's not from this planet. He just came here from another planet to help us in microbiology. He has a lot of very good tools. Right, so in this case, this time we will use the PROKKA. As you can see, there are different ways to install. So there are conda installation, brew installation, which you usually use on a Mac machine. There's a Docker image and also other images here and there. Of course, because now we have a working conda environment, we will do the conda installation. Before we do the installation, let's go out from the CheckM environment, because we don't really want to put anything else in. So if I start the conda installation now, I think we will. I think we are putting all the new files into the CheckM environment, and we don't really want to do that. We don't really want to make any collision or any problems between our newly installed program and the one which has its own environment. So what we have to do first is that we have to say conda deactivate. Yeah. And so we have to get back the base thing in front of our command prompt. And then just simply copy and paste the conda install line. This will take a few minutes, maybe, not too much. So the 2 main reasons you want to do genome annotation, and again, this is prokaryotic genome annotation. This is very important. I am not the good person to teach you eukaryotic genome annotation. That's way much more complicated. I saw a few times a few papers and I read about that, and it was always very it looked very, very difficult many times. In prokaryotic genomes, of course, we have the luxury that we don't really have too many uncoding or non-coding regions. We have usually an easily recognizable open reading frames and genes. We also know a lot of genes from prokaryotic genomes, so we can find genes much easier just by knowing them. So this is why it's much easier. And the reason why you want to do this thing, are you doing the same installation, Joe?
Yes.
Okay, so it's also solving the environment for you or something like that.
Yeah, it's also doing that.
Okay, so it will ask you to install all of these things as soon as it is able to. Oh my God, it will be a nice long installation because it's 500 megabytes, but let's hope that our internet connection at the server is fast enough. Actually, the downloading is not too slow, so I think it will be okay. Just feel free to start your installation as well as soon as you have the choice of saying yes or no. It will take time. Okay, so again, I'm starting the same sentence, I think, third or fourth times. So the reason why you want to annotate genes, first of all, because you are actually looking for genes in your in specific genes in your assembly, and instead of looking for specific genes by blasting them, you are just doing one gene annotation once, and from that moment you can see if that gene is there. If the gene is there, what is the sequence of the gene? So it's like finding all the genes in one step. Of course, because it tries to find all the genes. It won't be as sensitive or specific as a gene at a targeted gene search with, for example, BLAST. But it will be able to find most of the genes with very, very good reliability. And maybe that's your whole aim in your project, that you have a lot of isolates and you want to check virulence genes in them, you want to check MLSD, but you want to have the genes for the MLSD typing, or you want to see the antimicrobial resistance profiles, what kind of AMR genes are encoded, or you want to check if a specific metabolic pathway is present, and you have all those gene names in your list you have to find in your genome. So that's one of your reasons why you want to do a prokaryotic genome annotation. And I will tell you the other one as soon as we are starting our annotation. Did you finish?
Yep.
Okay, perfect. Because I think also the annotation process is maybe a few minutes, so I can just tell the remaining parts of the tale why we are running that. So we are going. If it's not too long, then everyone else can do it. Let's see how long is it. So let's go back to our stretch directory and to our own directory where we have the context FASTA. So we have to have the context FASTA here. This guy is not just making very nice programs and not just making them very easy to install, but also all the programs are actually very easy to use. There are always special options you can use. There are always special ways you can run something, but if you don't really want to bother about all these specific options, you can just simply run the program like saying PROKKA and context FASTA. And that's all. It will run, create the folder and do all the things for us. I think it is quite fast, so we can just run it together. Actually, it's using all the 4 cores. It will be some bit of it. It's not too big. Oh yeah. Now the server is a bit nice. Yes, I'm using BLASTP, but you are also using Perl, and we are now using BLASTP together. So it's also blasting protein sequences as well. So the other reason why you want to do the PROKKA is because you have a lot of isolates, for example, a lot of Salmonella or Staph aureus isolates, and you want to compare them. And if you want to compare them, you can compare the whole genome. And this is what we will discuss on Thursday, for example, when we will do the way how you can compare whole genomes by their SNPs. But you can also compare genomes by using the coding sequences, the genes, and only looking at the differences between genes. Of course, there are pros and cons again for both of them. If you are doing a core genome alignment, which means that you have 100 isolates, you have 100 assemblies at the end, and you are only comparing those gene sequences that you can find in all of them, or at least 98% of them. This is what we can call as a core genome. It is core genome because all of your isolates are having those genes. Then you will looking at a probably more conserved part of the genome. So you will probably see a bit less differences, but these differences are probably reflecting to changes in time in a bit better. So if you have different lineages in your species, then probably all of these coding sequence differences are accumulated better with time. But for example, if you have a subgroup in your 100 isolates, which are phenotypically a bit different. So for example, you found out that that 10 of these 100 isolates are resistant to specific antibiotics or are very happy to grow on a specific agar or a specific nutrient. Then there is a chance that it is not because they have a nucleotide difference in one of the core genes, but because they have some extra genes that make them capable of resisting antibiotics. This is almost always the case with antibiotic resistance, and they have that kind of AMR gene, for example, or they have some other metabolic pathway genes within their genome or within or on a mobile genetic element, and this makes them different from all the others. Mine is finished. It's actually not extremely fast. It's about 3 minutes. So if anyone else wants to do it, just feel free to run your own PROKKA. It is finished for me, and I think it is also finished for Joe. So feel free to run your own PROKKA if you want. You have your context FASTA file. If you don't have your context FASTA file, then copy it from the assembly folder. No, it's not there anymore. Let me just copy the context FASTA file into the main Scratch directory. So if you don't have the context FASTA file in your own folder, just simply cp:/context FASTA and dot, which means that you are copying the context FASTA from one folder up to your own folder. Let me just grab a glass of water. Okay, so if I go back to my previous tale about those isolates that are phenotypically different from the others, then probably, especially if we are speaking about prokaryotic genomes, probably you have to look for extra genes, which are also called like accessory genome or the genes that are out of the core genomes. But this is again something what you can do with PROKKA and PROKKA annotation. You just have to use the next proper tool. We won't speak about RORI today, but we will use RORI later, and we will speak about how you can compare different genomes to each other. And PROKKA is also very useful to extract accessory genomes. And then we will use some kind of visualization tool to see if you have a cluster, if you have a phenotypic cluster, then how you can find out what are the genes that are also there in that specific cluster. Okay, so as soon as you finish with the PROKKA, you will have a directory that is called PROKKA 05 blah 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 blah. I can imagine that this is some kind of random number. No, it's actually the date. Okay, it's May 20. It's the American type of date. Which is of course a problem if you want to run multiple annotations in the same day, because then it cannot really put into the same folder. So that's why I said that this is just the very basic usage of PROKKA. Probably you want to use at least 2 other options. And if you go to PROKKA, this is what you can see here as the moderate level of usage. So you can use the --out dir option and then give the directory name. So instead of having PROKKA underline and the date, you can just give your own name for the output directory, and also you can give a prefix for the files that are created, because if you look into this folder, then all the files have the same kind of file name as the folder. So you can define the folder name, and you can also define this file prefix, which is before all the extensions, by the --out dir and the -- prefix. So I think this is almost like the gold standard way to run PROKKA, but also if you just do this on one assembly, then it's okay. As soon as you did this, you can use Artemis to visualize your results, but we won't do Artemis today. But I will tell you what you can find inside. Maybe you are already familiar with a few of these files. I won't go through all of them. I think the most important ones. So the first one is GBK. I think this is something what you already saw probably, which is a gene bank file. So I think you already saw something like this. So you can see that there is a coding DNA sequence from here to there, which is probably the encoding the LSPA gene, which has this protein sequence, and so on and so on. There is already an error here, which is a problem with GBK5 format. When we spoke about text files in the very, very beginning of our course, I told you that there are some databases or data types that are using position-specific places for fields within the file. So if you look at this first line, you can see that in the very first line, there should be the identifier of the contig, but there should be the size of the contig as well. we know that the size is this because it is also written in the FOSTA header. And we also know that the size is here, but if you use this file and give it to a program that can understand the GBK format, it will drop you an error probably, because it doesn't really see the space between these two fields. There has to be a space. But the problem is that it's also here because it has to start in this position according to the standards of the GBK5 format. So a few programs will extract the size of the contig or this nucleotide sequence from here. Others will extract because they will just split the first line by the spaces, and they are looking for the third field, which is here only BP, but it should be the size if the contig FOSTA identifier would be shorter. Oh, sorry, yeah, this the whole 44, so it's a half megabase. So I think that there is some kind of option with the PROKKA as well, within the PROKKA, to make it more compatible with the GBK. I will try to find it. There are some extra options as well here, but maybe the best way, if you want to do a GBK compatible PROKKA annotation, is to truncate, to cut back the FOSTA identifiers. So just make a very short script. If you want, we can do this next time, a short R script that reads in your original file. So if you look into your context FOSTA file, you can ask the script to truncate all these lines and go back to like 10 or 15 or 20 characters. You will lose a few things. You will lose the coverage value and you will lose the length, but it's only interesting for you until you are doing your QC. So if you, for example, if you want to see what is the average coverage on your contigs, up to that point, you want to preserve this information, but after that, you don't really need that. So if you just simply write a short script, and again, if you want, I'm happy to write it together with you in R, and you see that let's truncate all of them up to this point, for example, or just up to this point, because this is also unique, because all the nodes, all the contigs are named differently, then it will be GBK compatible. If, of course, if you don't want to make it GBK compatible because you want to use another file format, I think one of the good file formats is, which is much easier to handle, is the GFF, as far as I remember. Yeah, in the GFF file, there are some headers here, but you can easily, again, you can easily just throw them away because they have a double hash mark in the beginnings. So if you make your own script, it's very easy to work with only those lines that are not starting with double hash mark. This is one line here, from here up to here. Of course, it is wrapped because of my window size. But what you see here is that within this sequence, from this position up to this position on the positive strain, so it is going with the same direction as your contig is going. It's not a reverse oriented gene. And starting with the 0 frame, I think we can see all the meanings of these columns. The GFF5 format is a very well known file. I think Artemis is working with GFF as well, as far as I saw here. Yeah, so if you are doing visualize in Artemis, it will read in the GFF as well. And then you have other informations, for example, the gene name here, some kind of function, maybe here lipoprotein signal peptidase as well. And of course, if you don't have a known function, I don't know if we have any hypothetical protein here. I'm pretty sure, yeah, for example, here you can see there is no real gene identifier, just a locus tag here, and the product is a hypothetical protein. So this is something which wasn't annotated completely. There is a potential gene there. There is a nice open reading frame. There is something which seems like protein coding sequence, but PROKKA was unable to identify the gene. And I think there is maybe the TSV. That's been the last one, which is interesting. Of course, in GFF, you don't have. Maybe you have the sequence at the end. I don't know. I'm not sure. But also you have a TSV, so you have a kind of tabular format of all of the genes. If you're just looking at your set of genes, because you are, for example, looking at beta-lactamase, or you're looking at some kind of other antimicrobial resistance gene, then maybe this is the easiest table to handle. You can easily read into Excel. You can look for your gene names or in descriptions. I think this one has actually a beta-lactamase. Yes, not one, but already more than 3 different beta-lactamase genes in this Staph aureus. So this is, of course, something which will make this isolate or this bacteria resistant to beta-lactam, to a few beta-lactam antibiotics. Okay, so these are the main files I think you would use. But as soon as you are using Rory or any other programs, just leave all the files here. The good thing with the novo genome assembly is that as soon as you are at your context FASTA phase, and all the post-processing you are doing, they will never really take too much space. So I think if I'm just checking the size of this folder, the PROKKA folder, the whole folder is 29 megabytes, which is nothing compared to the pre-assembly folder. I don't know if we are just looking at the files here, we have the deduplicated FASTQ files. We have the merged and unmerged files from Flash from last time. These are huge, several hundred megabytes. Oh yeah, this is something what I have to write into your cheat sheet as well. I haven't done this, so let me just remind myself that the cleanup has to be here, and I will write what are the cleanup steps. So what are those files that after you have a successful assembly, you can easily delete and free up the space. Right, and the last one, which is only here because it's quite practical for most of us who are here today, is a bit more specific program to find genes. So in many cases, you probably don't interested in the whole genome annotation, but you are much more interested in those things that are maybe clinically or medically relevant. So the same guy has another tool. And again, I will ask Joe to help me installing the program for the bioinfuser. So if you go back to the main page of this guy, then here we have the Abricot tool. And Abricot is using a lot of different databases to find antimicrobial resistance genes, plasmids, and virulence factors. So it's a kind of like a dense targeted search for important things. which is of course, again, I'm just doing or showing this to you because I know that many of you are interested in the same thing as I'm interested in. But I can imagine that there are other microbiologists all around the world that actually they are not really handle these genes as important, and they are much more interested in other genes. I think also for the Abricot, and also if we go back to the PROKKA just for 1 second, for PROKKA you can also make your own HMM databases. It's not as easy. It's a bit complicated, but if you know that there are, for example, genes that are always identified as a hypothetical protein, but you know what are those genes, then you can create your own hidden Markov model pattern for those genes and ask PROKKA to include your custom HMM profiles as well during the run, and it will annotate those genes from that point much better as well. And so it's a bit more complicated because you have to build up that HMM profile. It is much easier with the Abricot because I think it's somewhere here again, very easy to follow actually. Here you can update your database, but making your own database, this is the section. So if you have your genes of interest, because you are, for example, interested in a specific metabolic pathway, you can also ask Abricot to find those genes in your assemblies a bit more targeted and more specific way than and also it's much faster than doing it with PROKKA. I think it's very easy to follow, but this time we are only using Abricot to find those genes that are built in, those databases that are built in. So again, I ask you, Joe, to go down to the installation part. And again, we are going to the Bioconda one. So we will install this one. Let's go back to our home directory again. We are installing everything there. And just paste. I'm pretty sure it will be much, much faster this time, even if we have to download a few databases, but we don't have to download that many program modules or dependencies potentially. Is there any question for the PROKKA? Or anything else? Why we are waiting for the installation? There is nothing much to say about how this program is running. It is looking for gene sequences. So yeah, it's only this very short installation. I think, is this everything? It is also installed all the databases. That's interesting. So in this case, we can also check and we can also list what we have. So just feel free to do this, Joe, if you finished. Check if everything is okay. So if you say Abricot -- check, then it will look for all the dependencies. So it will look for BLAST_N. So BLAST_N is the nucleotide BLAST version of BLAST. I mean the command line version. So when you want to find nucleotide sequence against nucleotide sequence, BLAST_X is the one where you are looking for, you have a nucleotide sequence query. No, you have a protein sequence query, but you have a nucleotide sequence database. So that's the BLAST_X. Make BLAST_DB is just a BLAST database creator command, a few zip programs, and any2 FASTA. I don't remember what any2 FASTA is there. Probably just a converter program to make FASTA files, I can imagine. And the other one is -- list, which will give you the list of databases it uses, together with the sequences and together with the database date. This is very important, because if you install Abricot today, but you also want to do something next year this time, probably many of these databases, if you just think about ResFinder is updating, I think, like monthly or 2 monthly basis, because there are so many new antibiotic resistance genes. Also, there are many new plasmids we know. So it's always worth to update your database before you are running Abricot again. Of course, because we just installed this, these are probably all the brand new databases. But if we are not sure that these are all the new ones, then we can just simply do the update step here. So if you think that there is a new NCBI, for example, you can just do this. But I can imagine that we don't have anything new. Nice. And then it's updated. Yeah, so actually it is now a bit newer. I don't know if we have more genes. So we have now 5,448, and we had, oh my God, I cannot go back. Do you have the old numbers, Joe? Did you update?
Download it, so.
Oh, you did the update as well. Okay, perfect.
No, I haven't done the update. So
okay. So if you do the Abricot -- list, how many genes you have in your NCBI line?
NCBI.
5386.
Yep, that's the one.
Okay, so it's actually better now. So I think it works to do. I mean, if you want to do it automatically, again, we have 2, 4, 6, 8, 9 different databases. So what you can do again, if you can just make a very short script, it can be actually a bash script as well, where you put in all these lines for all the different databases and run it once at the same time, and it will update all the databases just with one command. The way how you can do this, I mean, I can actually show this to you if you want. So if I'm just going to my Scratch folder, so you can always, if you have multiple command line commands, and you know that you will run those commands after each other, always you will do the same kind of order and the same kind of commands. You can put them into one file and run that one file instead of running all of these commands separately. So the way how you do this is you create a new file. Let's say a nano Abricot databases update.sh. If you are always using a bash command line, you don't have to, but it's a good practice to start the script with saying this. Just check. Yeah, it's there. So this will tell the program that it has to use the bash as a programming environment, and you just simply copy and paste your lines here. What I will do, I will paste here this 9 times, and I will rewrite the --db for the different file names, which is a bit of So this is the database name here. So you have to use this E. coli _ VF. The second one will be the NCBI. I just want to follow the same order because it's a bit easier. And then the card database again, an antimicrobial resistance gene database. And let's finder. And then PFTP. This is probably a virulence factor database. I don't know it. Other antimicrobial resistance gene database. No, plasmid finder there. Plasmid finder here. This one and the last one. So this is how your whole script looks like. And what will happen, it will run all these command lines after each other. So it will run the first one, wait until it finishes, it run the second one, third one, and so on. With Control O, you can save, and with Control X, you can exit. Don't worry if you are not able to write this down at the moment. It will be in my folder. If you want, I can just copy out to the main Scratch folder, or you can also look at this. I'm sure that you can see my folder. At least, yeah, you have the reading access, so you can always see the LIOS folder in Scratch if you want. There is only one thing to do before we are able to run this. We have to make it executable. So as soon as you can see, this is our SH program here, and there is no X flag on it. So as usual, we have to do chmod u plus X abricate database sh, which will make it green, and it also puts this X flag here, which means that it is executable. And from this point, you can just simply say this. I don't really want to do it now because I don't want to spend the time with this, but if you want, you can run this script for the bioinfuser and see how many new genes will be there in your updated database. But again, it's much more interesting to run this program. And it is, again, because it's the same guy is doing installation and running very simple. So how to start? It is just simply, you just have to say abricate and then the FASTA file, abricate and context.fasta, and that's all. And what you will get back at the end, a list of genes that were found, which is here. Again, this is a tab separated txt file. So if you want to put this into Excel, for example, then just simply redirect the standard output and say abra output.txt or anything what you want, a file name, and then you can just simply download this txt file and read it into your Excel. Okay. Of course, as you can see again, we get back all the blah genes. So here we have those 3 blah genes that were found by PROKKA as well, but we also have a Mackey. This is something that why Ibrahim is sequencing Staph aureus genomes, but also some other antimicrobial resistance genes. Okay, this is the Abricot, and I will stop the novo assembly pipeline here. Of course, there are many other ways you can analyze your final assembly, but these are much more, I think, study or biological question specific analysis pipelines. And this, I think, this is covering most of those general ways how you can handle prokaryotic genome assembly. And before I just give you some hints on the de novo assembly and realignment, which we already spoke about that last time, but I just want to give you a few extra information before we start doing it on Thursday. If you have any questions now, or if you want to run any of these programs, even CheckM in the background, just feel free. Always check if someone else is running. If you see top and there is nothing running, there is no CPU usage, just feel free to start your runs. As I said, the document will be up there, so you can come back and do it anytime. All the command line comments are the first things here. So if you see it between quotation marks, of course, you don't have to put the quotation marks there, but these are always the command line comments. And of course, when you see something like path to mate1 FASTQ file, don't write in path to mate1 FASTQ file, but give the path into the mate1 FASTQ file, which is, the one of the pairs of the FASTQ files. Is there any question here? I also try to involve a few of these troubleshootings or QCs. So what are those things that you have to look for in each step, especially in the pre-assembly steps? So how can you become suspicious that something went wrong during the library preparation or sequencing or during your pipeline, and what you can do? Okay, if there are no questions, then I will only ask for a few more minutes. I won't fill up the 2 hours today. Thanks very much for Joe. Now, I hope that you are not that disappointed that you were last time when we struggled with all these installations. This is how it should go. So this is the normal way how bioinformatics software usage should go. Then you only have to copy and paste the installation command and then start using the program. There are a few times when it's not as simple. So before we go into the whole genome realignment, I was thinking about giving you a few decisions, and I think I will make some kind of flow chart about this for next time. I won't discuss this again, but it will be there, and it will be there if you want to look. It will be up in the Google Drive. So when you get back your whole genome sequencing data, you have these two choices. If you want to build up your own genome by using de novo genome assembly, or if you want to build up your genome based on a known reference sequence. As soon as you are doing eukaryotic whole genome sequencing, I think the de novo assembly is not really an option. Of course, if you are sequencing something that is not known and you have no chance to have reference sequence, you have to do de novo assembly. So, for example, when we analyzed Claire's fungus genomes. They found some interesting fungus on hedgehogs, and we got back the sequencing data, and we had absolutely no idea what they were. Then we did the novo genome assembly, but it's also true that they are very simple eukaryotic organisms. We found out at the end that actually the genome size is not way much bigger than a prokaryotic genome size. I think they had some kind of like 20 megabase genomes, which is is like just like 4 or 5 times bigger than an average prokaryotic genome. But as soon as you are going more up to more to much more different genomes, like mammalian genomes, and you have gigabases, it's very hard to make de novo assembly from whole genome sequencing. And actually, there is not really a reason to do so, especially if you know the reference sequence. If you have any kind of maybe not perfect, but some kind of draft whole genome sequence, then you have something to compare to. The reason why realignment is working very well with eukaryotic genomes is that even if you have big structural changes, sometimes in eukaryotic genomes as well, most of the cases these huge changes are not coming from outside. So what I'm saying is that if you have a huge deletion or insertion or chromosomal translocation, which are really huge changes, one arm of a chromosome is breaking off and going into another arm of a different chromosome, which is quite often happening, for example, in cancer cell lines. There are usually quite a lot of huge genome rearrangements. But even if something like this is happening, you don't really introduce new DNA sequence into the genome. You are just having rearrangements. You just have to find out, okay, something went really wrong because the sequence which I had in this position in the reference sequence before is not here anymore. It is there somewhere else, but it's not here anymore. So you have to find out how the genome rearranged, or if you have an insertion. In many cases, in eukaryotic genomes, you can have a lot of big insertions as well, but it is usually happening like a tandem duplication. So you have, for example, ALU sequences that are a few hundred base pair sequences that are very homologous to each other, and during the recombination process, when the meiosis is happening, there can be a situation where the whole crossover is not happening symmetrically. So it's not the two pieces that are exchanged between two chromosomes are not the same thing, just because it is slipping, for example, on an ALU sequence. So what will happen is that you will have, for example, you had a gene with 8 exons, and what you will end up in your new cell line is the same gene, but with 9 exons, but only because exon 4 is duplicated. So when you resequence this genome, you will find out that you are having more than 1 copies from the same thing, but you already know the sequence because it is coming from the genome itself. And this is the huge difference between eukaryotic and prokaryotic genomes, or especially prokaryotic genomes and high-level eukaryotic genomes. High level means also, insect genomes or vertebrate genomes. That if you have any insertions or seemingly new genetic material, in eukaryotic genomes, the new genetic material is coming from somewhere else of the genome. But in prokaryotic genomes, it is very often coming from somewhere else, from a phage, from a plasmid, from another different mobile genetic element, a transposon, which means that if you are doing genome realignment and you are using your reference genome, but that reference genome doesn't have that sequence, that extra sequence there, then you will just simply miss that gene from your genome assembly. So that's why it is very important to know that if you are assembling prokaryotic genomes, if you really want to have all of your genes, you have to do de novo assembly. If you are not interested in those genes because you are just want to see the difference of your new sequencings, of your new isolates compared to your reference, then it's okay. It's okay to do realignment. And we will do this next time. And you can extract a lot of information from this, but you are also missing information. And this is very important to know.